Hey guys, it's Miss Willis, and today we're going to be doing our notes over the introduction to biology. So we're looking at the scientific method, characteristics of life, and then we're going to actually save biological themes for the next day. So looking here, how the notes are set up is that we will see all of the goals for each section outlined at the beginning of each section. So nature of science, we talked about this a little bit already of what makes something scientific. And the first thing is that it's empirical, which means it is testable. We can measure it. It is tentative. It can change over time. Sometimes we develop new technologies or we're collaborating more, and so we find out that it works differently in other places, but it's never 100% positive. Like, it can change. So that's what tentative means. You've heard of like a tentative schedule. Like, it could change. We're just gonna find out. Like when it says TBA in a schedule, like to be announced. All right. Observation versus inference. Observations are simply taking in information and stating it. So Miss Willis is wearing a tan sweater. Ms. Willis has her hair up. Ms. Willis is wearing her lanyard. These are all observations, things that are true that we can simply see. An inference would be when you take all that information and say what it probably means. So the inference could be Miss Willis is at school. Miss Willis is dressed for school. That idea. Or you could say, like, in regards to wearing the sweater, it is cold in Miss Willis's room. That would be an inference. You don't know it, but you're using the information that I have. Like if I was wearing a winter coat, you might assume that it's cold in my room. That would be an inference, taking the observations you have and saying what it probably means. Number 4, subjectivity. We all come in with our own background information, which is why a lot of times we can view information differently. So if I tell you the word ball, B-A-L-L, ball, I want you to visualize what you're thinking of in your mind when I say the word ball. Some of you are probably thinking maybe a bouncy ball, some sort of athletic ball. But actually what I'm talking about is a dance, like Cinderella went to the ball. So subjectivity changes where you might put information together because of the background that you already come from, the amount of knowledge you already have. It's collaborative. Scientists work together. We share our tests. It's not just like, ooh, yes, I want to be the only famous person. You have to share. Other people have to test it too in order to find out if it's truthful, if it actually works that way, or if maybe you were being somehow biased in what you, how you tested. And then it's peer reviewed. It is shared out amongst others, other people of like similar background knowledge, or not even just background knowledge, but like scientists with other scientists, not just like a scientist shares it with me. And then remember that science is limited. We talked about superstition and things like that, talking about higher power causing things to happen. That's not scientific. It's not to say it can't be true. It's just that it's not scientific. When we look at different branches of science, we have our living sciences, biological sciences, things like botany, which is the study of plants, zoology for animals, ecology, the environment, physical science, when we look at physics versus chemistry, and then earth sciences, about the earth. So looking at geology, studying rocks, meteorology, astronomy actually gets included with earth sciences as well. Then we get to the scientific method, and you guys are probably familiar with this. Just in general, scientific method, you have a hypothesis, you have an experiment, you have all of those parts. So looking at how all of that fits together, the first part is simply making an observation or a question. You look outside and make an observation and you wonder something. That's the first part of the scientific method. So kind of like the curiosity part, how did we even decide to do some sort of experiment. So that's the first part, is making an initial observation or a question. How does something happen? How does light affect plant growth? And then we can make an experiment to test that. So then we make observations. Make sure that your observations do not have your judgment in them. I don't know if any of you are fans of Teenage Mutant Ninja Turtles, but they are all about pizza not having anchovies on it. And so saying that pizza is disgusting would be a judgment. You can't say that as an observation. Or that pizza has anchovies on it. That is an observation. If we were together, we were going to do this just like a makeshift kind of lab, not like a serious lab, where I have different liquids for you, and you're going to be able to smell them, look at them. Some of them are different colors, and some of them don't smell very good. Your observation could be there is a strong odor from one of them. It can't be one of them smells nasty. That would be putting your judgment on it. And so our observations need to be without judgment, without bias. A lot of times the responses I get to that is that number 3 or whatever number it is smells like vinegar. That's actually an inference. You could say it smells acidic, it has a strong odor, but to say what you think it already is would be an inference and not an observation. So that's something to remember too. Then you form a hypothesis. We know that when we describe a hypothesis, it's an educated guess. Well, you have to have 2 parts. You have to have the guess and you have to have the educated part. So what you think is going to happen would be your guess, right? It might have a prediction of what you think is going to happen in the end result. And then the educated part is what led you to it. So like when I said earlier with light having an effect on plants, plant growth, I could say that a plant in continuous light will grow more than plants without continuous light, something like that. It must be testable. We can put a plant in light for 24 hours for 1 or 2 weeks, and that is testable. We can measure it. So make sure that it's something that's measured. We can't just say the sky is blue. We can't test that. Like it is an observation, but it's not like a testable, like that we can form an experiment over it. Then we get to experimental design. And so we have to have a control group, one that gets the normal conditions. So I talked about a plant getting continuous light. We know that the sun comes up, is out for however many hours, and then it goes away for however many hours. Let's just say 12 and 12 to make things easy. And so because of that, we need a control group, one that gets the normal conditions. So I'll have a light that's on for 12 hours and then it turns off for 12 hours. And then the experimental group has all of your different, there will be multiple parts within your experimental group. So you might have the ones that are in continuous sunlight. Maybe you'll have ones that get 3 hours of sunlight and then the rest darkness. Some that are in complete darkness. Let's just make it easy. So our control group was that they get 12 hours of light and 12 hours of not. Our experimental group's going to have 2 parts. The one that has a continuous light, which is what we're wanting to know about. And then we take the opposite of that, one that never gets any light. It's in complete darkness 24 hours for however long we're doing this for. Okay, so those are control groups and our experimental group. And our experimental group had a couple of parts within it. Then the variables are the parts that we are changing, that we're going to say that when we measure it, this is probably what caused it. And so variables in this case, what did we change from one plant to the next? How much light they get. One gets half and half, one gets full sun, one gets no sun. So the amount of light is our variable. In a controlled experiment, which is what we always want to do, we have one variable. Because there are other things that can affect a plant and how well it grows. We could talk about what type of soil it gets. We could talk about how much water it gets. But we want to just change one of those things in order to impact and say that this is what's causing the growth change for our plant. So very last part here, a controlled experiment only tests one variable. All others are held constant. Constant is different from control group. Remember, control group gets the natural conditions. things being held constant means that it's the same between the control group and the experimental group. And we're going to do more examples of these coming up. Then we get the independent variable. Think about it. Independent starts with the letter I. It is what I am testing. Okay? So am I testing the amount of light? Am I testing the amount of water, or the effects of water, the effects of light, the effects of soil? Whichever one, it's going to be one of them. And that's the independent variable. Then the dependent variable is what I use to measure the change. And what's cool about these guys is that this is also how you set up a graph. So when you have your X and Y axis, the independent variable is going to be going across the bottom, and the dependent variable goes along the side. So the X, it goes with your independent variable, and then the Y goes with your dependent variable. That should make sense, because if something is in more light, then we should see that it grew more. So the dependent variable's got to be what's going up that Y axis. And again, reminder from the last slide, constants are controlled variables. We prevent them from being the variable we are testing. So like with the plants, if we're testing the amount of light, then we want the soil, the plant type, the amount of water that they get. We want all of those to be the same, whether they are in the control group or experimental group. So those are constants, things that are the same or constant, consistent from the experimental group to the control group. So in our example here, we're going to look at what effect water has on plants. And I'm just going to start filling this in. So we're saying that normal conditions for this plant is that it needs 1 cup of water every day. We did some research to find this out. So which plant is in the control group? Oh, sorry. What are we going to do with the other ones? We're going to give this guy at the top half a cup of water. This guy's going to get 2 cups of water. This guy's going to get 2.5 cups of water. So which plants are in the control group? The one that gets the 1 cup of water, because we researched and found out that that's what is normal for it. Which ones are in the experimental group? The one that gets half a cup, the one that gets 2 cups, and the one that gets 2.5 cups. Then what things need to be held constant for all of them, whether they're in the control group or experimental group? We need the same type of plant. We need the same sunlight, the same amount of sunlight, the same water source. So if I'm pouring it out of the faucet versus using bottled water. The plant type, we can't compare the growth of a tomato plant versus a daisy. Temperature and the container that it's held in. All of those need to be the same. They don't need to be in the same container as each other, like all grouped together. But if I get like what brown pot that's this big, then that needs to be the same one that I use for all of them. So in our test here, in our experiment, what is the independent variable? What are we testing? Think for a second. Testing the effect of the amount of water that they receive. What's the dependent variable? How will we measure the effects? Well, it's a plant, so we will measure the growth. And so then we could form a hypothesis from this. If we give the plants this many cups of water, this would need to actually be full. These are just a couple different examples that you could test. If we give the plants 2.5 cups of water, then they will grow the most because the extra water will feed the cells more consistently. A different one would be the plants that receive 2.5 cups of water will experience the most growth due to excess hydration will nourish the cells more consistently. Anything like that would work. Then for collecting and analyzing data, quantitative would be numbers, like with our measurements. Qualitative would be descriptions. Maybe with the different things we're doing to the plants, maybe their leaves change color. They turn yellow. They turn completely yellow. The tips of them turn yellow. They start to lean over and wilt. Any of that would be qualitative. So quality, talking about descriptives. Quantitative, talking about numbers. And then drawing conclusions, saying what happened and making inferences from that. So saying that because this happened, this is probably what that's going to mean. Reaching a final conclusion and then publishing that, letting other people try it out too. We're not going to worry about theory versus a law of nature, and that's where we're going to leave for today. So There's going to be a little quiz on Canvas. And to summarize this, I'll see you next time.